Hello, everyone. Good morning, and thank you for having me here. My name is Hernán Hoyos-Maya. I'm a biology student from Universidad de Antioquia, and I'm going to present to you the identification of key angiogenesis and notch pathway genes related to CADASIL disorder in the gliovascular unit of mouse basal ganglia and human frontal cortex. So, to do a brief introduction about CADASIL. CADASIL stands for Cerebral Autosomal Dominant Arteriopathy with Subcortical Infarct and Leukoencephalopathy. So now you know why we call it CADASIL, because it's a really long name. And it's caused by mutations in the Notch3 protein that lead to a loss of the small vasculature in the brain and a degradation of the white matter of the brain, producing a series of symptoms in which we can find vascular stroke, seizures, and dementia, leading to lifespan shortening. And the Notch3 signaling pathway is a really important pathway in vascular cell type because it's a content-dependent pathway that depends on the Notch3 protein that gets translocated to its transmembrane location, and it interacts with the receptor cell in a ligand, and the intracell, it has two domains, an intracellular domain and an extracellular domain. The intracellular domain gets translocated here again to the nucleus, where it works as an enhancer of the transcription of genes related to the formation of new vasculatory cell types in a process called angiogenesis. And this promotes correct homeostasis in the brain, between the vasculatory and the parenchyma of the brain, leading to a homeostatic relationship between these two tissues. But in the CADASIL environment, there is an aggregation of this Notch3 protein because of its mutation inside the signaling cell, and this produces a deficiency in the Notch3 signaling, augmenting the stress in these cells, leading to apoptosis. And in this Notch3 deficiency environment, there are loss of these vascular cell types and an accumulation of waste products in the parenchyma of the brain. So the vasculature in the brain is composed by an endothelium surrounded by small muscle cells, and in the smaller vasculature, there are pericytes that are glial cells that are in the gliovascular unit of the brain. The gliovascular unit is where the parenchyma of the brain, the glial cell types, connects with the vasculature in the brain, and we have glial cell types like astrocytes, like oligodendrocytes, pericytes, and microglia. This last one, microglia, is really important because microglia works as the immune system in the brain, and it can detect pathogens, it can detect lesions in the vasculature, and can produce inflammatory signals to repair these problems in the brain. But in the context of CADASIL, this works different, because in CADASIL, currently there is no cure or effective treatment. And we have a state of hyperactivation of microglia, loss of neural cells in the brain, and an hyperactivation of inflammatory response in the brain. And we wanted to know which are the gene mechanisms in these cell types of the gliovascular unit in CADASIL that could lead us to a potential gene target that possibly can lead us to a treatment, a better working treatment for this disease. So to do this, we perform analysis in humans and mice models. For the human model, we perform single cell analysis in the tissue from the frontal cortex of the brain of CADASIL and healthy patients. Then we calculate the cell population in single nuclear RNA sequence, and then we perform analysis with pseudobulk to find DEGs in these clusters. Then with these DEGs, we perform a gene ontology analysis to find the process that these DEGs are related to. And finally, we perform a transcription factor analysis to see which of the transcription factor correlates with the gene expressing in these gene ontologies. And in the murine model, we use a Notch3 deficiency murine model that consists of a Notch3 knockout of mice, and we use tissue from the basal ganglia of the mice and perform single cell RNA sequencing analysis and the same downstream analysis as for humans. Then the results we obtained is first, for humans, we found expected cell populations that we could find in the frontal cortex of the brain. We found several neuronal cell types in here. And we also find gliovascular unit cell types like oligodendrocytes, astrocytes, microglia, and vasculatory cell types. And in mice, we found the expected cell types that we could find in the basal ganglia with a higher proportion of glial cell types like microglia, astrocytes, oligodendrocytes, and then we ask if there are some difference in between these cell populations between CADASIL samples and control samples. And we found for humans that there is an increase in microglia in CADASIL patients. This is a state called microgliosis that has been reported to CADASIL patients that correlates with a higher signal of inflammation in the brain. And in the murine model, we found that there is a loss of almost 50% of mural cell, vascular cell, endothelial, and smooth muscle cells in the knockout of the notch tree, maybe because of this knockout. So this is a model for CADASIL because it represents this deficiency in notch tree and also the loss of mural cells. And then we ask which are the DEGs that we could find in these different cell types we found. And for humans, we focus obviously only on the gliovascular unit cell types, and we found that astrocytes, microglia, and oligodendrocytes are the ones that have a higher number of DEGs upregulated in CADASIL. And also in the murine model, we found a similar pattern where astrocytes, oligodendrocytes, and microglia have a higher number of DEGs in the notch tree knockout. But now the question is, what cellular process are related to these DEGs? So then we perform a gene ontology analysis, first for humans, and we found process related to angiogenesis, inflammation, and notch signaling that were enriched in cell populations as astrocytes, microglia, and vascular cell types. And it's really interesting because in most we found a similar pattern where we have a higher signal in the angiogenesis, but in the inflammatory response, we found a higher signal in the astrocytes, oligodendrocytes, microglia, and in the vascular cell types as well. So this leads us to the question that there is an increase in the angiogenesis response in these DEGs, but also there is an inflammatory response. And we know that microglia is responsible for this inflammatory response in higher part. And we wanted to know if there is a transcription factor that is co-expressing, that is regulating the expressing of these DEGs related to these ontologies. So briefly, I want to talk about a transcription factor that is a gene that can regulate the transcription of that is a protein that can regulate the transcription of genes by binding to its promoter region. And we perform the analysis and see which of the transcription factor we found were expressed in which cell types of the gliovascular unit. And we found a lot of transcription factor related to the DEGs for humans and for mice. But we focus on one specifically, that is RONX1, that's RON-like transcription factor 1, because it only expresses in microglia. And this transcription factor is related with the proliferation and differentiation of vascular cell types and also the immune system cell types. So it is really interesting because microglia is like the immune cell type of the brain. But also we found the signal only in this microglia, and this transcription factor is related to angiogenesis. So maybe that is the transcription factor that is related to all these DEGs that are enhancing the process of angiogenesis and the inflammatory response. So now that we have this environment in CADASIL where hyperreactivity of cell types, now we know some insights on the on the molecular and genes expression in these cell types, and we found RUNX1 as a potential transcription factor master regulator that could lead us to a possible treatment. But this is what we want to prove in the future. So now we did a test to see if RUNX1 is highly expressed in the CADASIL patient compared with a control. And with this immunohistochemistry and immunofluorescence in a CADASIL patient and in control patients. And we found that in CADASIL patient, we found a higher density of cells expressing RUNX1. Also, we found a higher density on microglia expressing RUNX1, and also found a higher signal of RUNX1 in the vasculatory. That leads us to know that RUNX1 is actually highly expressed in the brain of CADASIL patients. And the take-home message is that we provide information about the gene regulation in CADASIL environment for human patient and a murine model. That gliovascular unit cell types of CADASIL tissue present enrichment of process related to angiogenesis, inflammation, and notch-related ontologies, and that we identified RUNX1 as a master regulator expressed in microglia and provide proof of concept evidence of this higher expression in CADASIL patients. And thank you for listening, and to all the people involved in this work, and for the opportunity. Thank you very much. We have some questions.
Very nice work. Have you also find something related with hypoxia or hypoanalpha or any consequence of it?
We haven't tried to find something like that, but because we were focusing mostly in angiogenesis and inflammatory response. But we could try to find something like that in the future.
Thanks for the right. I was curious to know, what do you use as controls for the human experiment?
We use a healthy patient that doesn't have CADASIL.
Then the problem is which tissue?
With tissue frontal cortex.
Oh.
Postmortem.
All right.
Of course. Sorry. Sorry.
With all of the difference that you have, is the mouse a good model to study this disease?
Mouse has been shown because we found the same that we found in humans in mouse. It's like it was almost the same. So it's like we have a second proof that it works in human, but we found the same thing in mouse.
Well,
So
the last transcription factor, but all of the different processes were like not so similar.
not so similar, especially in the cell population, because we use different tissues, but in the ontologies and the transcription factor analysis, it worked pretty much very similar.
So great.
Thank you.